In this video, I'm just going to go over some of the common metabolic tests and talk about their results. So here we have the PR broth. If you recall, PR stands for phenol red. Phenol red is a pH indicator that turns yellow in the presence of acid. Acid is typically produced when a microorganism is going to be utilizing the sugar that we put in here. So PR broths come with all different types of sugars. You order them with a specific type of sugar, so mannitol, glucose, lactose, sucrose, those types of different types of sugars. So if that sugar's in the medium and the microorganism has the capability of eating that sugar, it's going to produce that color change. So this tube starts off red or pink in color, and then it turns yellow in the presence of that acid. So here you can see that it's yellow. This is a positive result. And then you can also see this inner Durham tube. A Durham tube traps gas. So it's an upside down tube that's designed to trap gas that's produced by the microorganism. If this microorganism produces gas while it's utilizing that sugar from metabolism, then you're going to see gas accumulating in this tube. So this is positive for gas. You see this gas bubble. If it was negative for gas, then this water level inside the tube would be all the way toward the top. The Catalase test. The Catalase test tests for the presence of the enzyme catalase. Catalase is an enzyme that can break apart the toxic hydrogen peroxide that can be produced as a waste product or byproduct from cellular respiration. So hydrogen peroxide is a bad guy. Cells don't like it. If a cell is able to use oxygen as a terminal electron acceptor in that electron transport chain, chances are it's going to end up producing this hydrogen peroxide. And if it produces it, it's got to have a way to break it down. So it's also going to produce that catalase enzyme to protect itself from damage from that hydrogen peroxide. So here are two different samples, and you can see in this test, you smear bacteria on the surface of the slide, and then you add hydrogen peroxide to the slide. The presence of bubbles is a positive result. Bubbles means that the enzyme catalase is present, and it's breaking apart that hydrogen peroxide to release a gas. If there are no bubbles, that means it's a negative test result. The oxidase test. So this is a positive oxidase test. What you do with the oxidase test is you swab it on some bacteria, and then you put those drops of oxidase reagent on it, and a dark purple or blue color change is indicative of a positive result. There are also cards that you can use, and those typically tend to turn more blue in color than purple, but nonetheless, we're looking for the presence of cytochrome C oxidase, which is an enzyme in the electron transport chain. So if it's present, then it's positive. So here you can see a positive result. And now we get into the Invic. So the Invic, I is for indole. The M is for methyl red. The V is for Voges-Proskauer. And the C is for citrate. So when we report an Invic score, we're going to report it as a positive or negative result for each one of these. So looking at this series of tubes here, they're in random order, and there's a couple extra tests in here, which we'll discuss in just a little bit. But when we want to report the score of the Invic, we're going to first look at the indole test. So the indole, they say that they did a spot indole test. Spot indole, you would smear some bacteria on the surface of some filter paper, and then you add the DMACA indole reagent, and the presence of an aqua blue color change is positive. So it's testing for the indole that's produced when the amino acid tryptophan is broken down. So if tryptophan is broken down, indole is produced, and we can detect that using that indole reagent. So here we can see a positive indole test. So for our Invic score, we start with a positive. M is for methyl red. So we grow it in this MRVP broth, and then we're going to add methyl red to it, and if it turns red, then it's a positive result. So we are going to mark this as positive. And then the V is for VP, or Voges-Proskauer. Voges-Proskauer is looking for acetone production. as a byproduct of cellular metabolism. So we're going to add a couple drops of aponaphthol and a couple drops of potassium hydroxide, and we're going to look for a red color change. This is not red, so we're going to mark that as negative. And then our citrate. So citrate is sugar that's in the medium, and if the microorganism can use that, it's going to eat up all the sugar, and then it's going to start to produce a base. So it's going to use the ammonia in the media and produce that base. And if a base is produced, it's going to produce a blue color change. So a positive citrate test would be a blue color. Green would be negative. So this is green. It's going to be a negative result. So what we end up with here for our Invic score is positive, positive, negative, negative. Then we can also look at phenylalanine and TSI. So phenylalanine is a pretty big amino acid, and if microorganisms have the ability to break it apart to use it in the Krebs cycle for energy production or what have you, then you're going to see the growth on the phenylalanine slant. And when you add ferric chloride to it, so a couple drops of ferric chloride, you're going to see a dark green or gray color change. Here you can see it's yellow. This is a negative phenylalanine test result. And then we have the TSI slant. So the TSI, triple sugar iron, this tests for the ability of the microorganism to use certain types of sugars, and then also tests for the presence of H2S production and gas production. So when we score a TSI slant, we're going to do the slant over the butt, and we kind of divide it right here. In the bottom part of this, we have glucose, and in the top, we have sucrose and lactose. A is for acid. K is for alkaline. So if we see a yellow color change, then we know that that was an acid produced, and we're going to mark it as an A. If there is no color change, if it stays pink like the original color that it came as, then we're going to mark it K for alkaline. So how to score this would be A over A, because both the slant and the butt, or the bottom of it, are both yellow. And then you can see some separation here at the bottom of the tube. There's some gas that was produced. So gas was produced here. We're going to put plus gas. And that would be our result for our TSI slant. Here's another example here. So I just explained all the tests. I'm just going to read the results right now for this. So if we want to get our Invic score, we look at the indole, which is negative. And then we look at the MR, which is positive. And then we're going to look at the VP, which is negative. And then here our citrate, which is positive. So now our Invic score is negative, positive, negative, positive. Our phenylalanine is a negative result. And our TSI is A over A, H2S positive. You can see this black color change here indicates that H2S was produced. So H2S positive for this TSI slant. Now this one, we're also going to score the Invic. So Invic, we have a negative, positive, negative, negative. We look at indole, MRVP, and then citrate. For our phenylalanine, we actually did get the color change. So when we added this, we got the color change. Here we have a positive phenylalanine test. And for our TSI slant, we have A over A, H2S positive. Now this is our starch hydrolysis test. Starch hydrolysis, we put starch in the plate, and we're looking for the presence of the enzyme amylase. If the microorganism produces the enzyme amylase, then it's going to be positive for the starch hydrolysis test. So what we do is we're going to streak 2 lines of growth down the plate. Sometimes we'll use 2 different species, like in the case here, and then we're going to let it grow. After it grows, we're going to douse the plate with iodine. Iodine is going to form a complex with the starch. And if the starch is present in the agar, it's going to form this dark color. So you can see starch is all over this agar here, and it's all around this microorganism over here. But what happened here is the microorganism had the presence of amylase and broke down the starch. So there is no starch in the agar. There's nothing for that iodine to form a complex with. So you can see a clearing around the growth. So bacterial sample A is positive for amylase. So this is a positive starch hydrolysis test. And B is negative, so it doesn't produce the enzyme amylase, and you can see that there's no clearing around the growth. There's no starch that was digested, so we have iodine forming a complex with the starch that wasn't digested around that microorganism. Now for this one, we have the milk agar plate. Milk agar contains milk, so it's usually skim milk agar, and it has that milk protein casein in it. If the microorganism produces certain proteases, proteases are enzymes that digest proteins, you're going to see some clearing around the growth. So here you can see clearing around the growth, and this one is positive for protease production, and here you cannot. So here it is a negative test result. The urea slant, if urea or if the enzyme urease is produced by the microorganism, it has the ability to break down urea into ammonia, which is a base. That base is going to cause a color change in the medium to this pretty hot pink color. So this is a positive urea slant. Any other color, it was originally like a yellow, orange, or red color, and then it would be like a yellow or orange, or any other color other than hot pink would be considered a negative result. So here are just some more examples of TSI slants. And recall that we mark the top part of it first. So I'm just going to give the results for these. This first one is going to be K over K because there was no yellow color change and there's no H2S produced. So K over K. This one is A over A, acid over acid. It's all yellow. This one is K over A, H2S positive, because of the black color. And then this one is K over A. So that's how you would score all of these TSI slants. So hopefully this was helpful in going over some of the major metabolic tests. You can watch the other videos and take a look at the tests being performed.